Okay, so good morning, everybody. It's my pleasure to introduce Dr. Eva Brewer. She is a resident in our clinic. I think most of you know her. And since 2019, she came to the lab to perform her PhD. And she is working on the liver regeneration, as you see the title, a pretty long title. She will read the title herself. She is a very curious scientist, and she has a great flair for scientific techniques. So we are very happy that she is in the lab, and she will give you a quick snapshot of her research project. Eva, please.
Thank you very much, Anurag, for this nice introduction. It's a pleasure to give you a short insight in our research. My project is about zonation in the liver during liver regeneration. So what does each hepatocyte during the regeneration after hepatectomy? So let's start with the liver. We all know the liver is our central metabolic organ in our body. It has many different functions. It stores and regulates glucose and fat. It produces amino acids. It has many detoxifying functions. And it has one unique thing about the liver. It can regenerate. So we can take away about 70 to 80% of the liver tissue, and the rest will grow back to its initial size within weeks. This is unique. But there is one thing. What if we take away too much tissue? Then we have a problem, because the remaining liver parts will not be able to regenerate. And we have something called post-hepatectomy liver failure. Many patients that, for example, have not a good liver, have a fibrotic liver before, are at risk to get post-hepatectomy liver failure after hepatectomy. Our goal is to get a better insight into liver regeneration and to get a better understanding on how the liver regenerates to overcome also these problems. And this is the reason why we're doing this research. So how do we do this research? So I just want to get a little insight into our mouse model. So you know there are, in the meantime, organoids, we have organs on a chip, but at the moment we don't really have a model that does not rely on the animals. So we have our mice. This is a mouse liver. It looks a little bit different to our liver. So we have different lobes. We have the middle lobes, we have the left lobe here, we have the caudate lobes, and we have the two right lobes. And depending on how much or how many of these lobes we take away, we have different forms of hepatectomies. So this is the liver in situ of a mouse, and our standard hepatectomy model is a 68% hepatectomy model, where we take away the middle lobe and the left lobe. This looks like that in the animal, and you can see that we really try to take away most of the tissue, of the remnants, or of the segments we take away. On the other hand, we have the extended hepatectomy model, where we take away the caudate lobe and the right anterior lobe, and in situ this looks like that. So actually what is left is only this right posterior lobe, and this is sufficient for most of the mice to survive. And we know that after standard and also extended hepatectomy in mice, the liver will grow back to its size within a week, approximately. And you can see here, this is 2 days after hepatectomy. The remaining lobe has already gained in liver weight, and you can also see that it's a little bit fattier than before. If we take a look at the regeneration, that's what we know so far. So after standard hepatectomy, where we assume that all animals will survive, all will regenerate, we have an entry into the cell cycle, which is very orchestrated. And approximately 2 days after hepatectomy, we have a peak in mitosis. So many cells are in mitosis 2 days after hepatectomy in mice. Contrary to that, if we take too much tissue away, we know that we have an S-phase entry, so cells start to get into the cell cycle, but we see less and also delayed mitotic peak. About 20 to 30% of these animals will die due to this operation, and all of these animals will develop some sort of jaundice, some more, some less. On the other hand, and this is important for my project, if we take away just a little bit of the tissue, so for example, a minor hepatectomy where we take away about 1/3 of the liver, we don't see liver proliferation in the sense of cells dividing. But what you see is hypertrophy. So the liver will also regain its weight, but not by dividing the cells, but mostly by hypertrophy. So one cell gets bigger. And this is more or less the basis of my research. My question is now, how can the liver regenerate and at the same time provide its vital metabolic functions? So we know we can take away the tissue and it will regrow and it will grow back. But at the same time, the body needs the function of the liver. And how does the liver do that? To get a better insight, we have to take a look at the anatomy of the liver. So the liver consists of thousands of little liver lobules. And each lobule has a central vein in the middle and portal veins on the outside. Here we see that the blood flows from the portal vein to the central vein. And along this portocentral axis, we have a lot of hepatocytes that look more or less the same. But each hepatocyte has, depending on its location along this axis, different metabolic functions. So we have the periportal hepatocytes, we have pericentral hepatocytes, and we have these midlobular hepatocytes. And we know that about half of all the genes and the encoding proteins are zonated. So we know that half of our liver functions is zonated. So it's either periportally expressed or pericentrally expressed or somewhere in the middle. And what is responsible for this zonation is not really known. So we assume that the partial pressure of oxygen is an important factor, but it is not the only one. And the second thing is that this zonation is not something static. We know that it can be changed. It's a very dynamic thing. So zonation is known for quite a long time and is well researched also in disease models. But for example, in regeneration, only in the recent years, it was possible to get a better insight into this topic. So what a former lab member of our lab saw, and which was also kind of the starting thing or the starting point of this project, is that she observed that 2 days after hepatectomy, there was a difference in cell size. So what she saw is that the hepatocytes next to the central vein were much bigger compared to the ones next to the portal vein. And she saw also that the mitosis were more around the portal tract, so the portal vein, compared with only less mitosis around the central vein. And this goes hand in hand with a faster restoration of glycogen around the central vein. So she assumed that the central vein is the area where we have hypertrophic cells, so bigger cells and cells that provide the metabolic functions, while the cells around the portal vein proliferate and actually divide in the end. So also during regeneration, the liver seems to be zonally controlled. But what is this hypertrophy? There is a very interesting paper from the Schiebler group where they saw that the liver size and the size of each hepatocytes changes in a circadian rhythm. So during the night when the mice are feeding, the hepatocytes grow in size, and during the day when the mice are sleeping, the hepatocytes get smaller. And this accounts for about 10 to 20% of the liver weight, which oscillates in this circadian rhythm. But this was the idea, okay, if we have these big hepatocytes, they were able to connect this to ribosomal content, to the cell size, and also to the protein content. And our hypothesis was now that we have, during the liver regeneration, we have proliferating cells, which are needed to restore the liver mass in the end. And we have also hypertrophic cells, which are needed to get the liver functioning during this liver regeneration. And the problem is, if we take away too much tissue, the liver is just overwhelmed with these metabolic tasks and simply gives up. How do we do this research? We have, as I said before, we have these different animal models, and this is, for example, the standard hepatectomy, and we operate these mice, and then we take the tissue and analyze the sera and the tissue after several time points after these hepatectomies. Standard, but also extended hepatectomies. And one of my starting projects was to look at the co-localization of certain events, so the proliferation, but also metabolic functions. And for that, I used multiplex immunofluorescence. For example, you maybe saw fluorescence pictures, and it is possible to now kind of stack several different proteins or stainings on top of each other to see what proteins are co-localized to each other. And I wanted to see, are there changes in the zonation during liver regeneration? Is proliferation also zonated? Are these metabolic functions zonated? From some, we knew they are expected to be zonated, but we don't know what happens during proliferation. And are there changes in the zonation during the regeneration? So this is one of the stainings. Here we see the entire slide. And for this type of multiplex stainings, we need a special microscope, a special software to overlay these stainings and to make kind of a spectral, to get rid of the spectral overlap of these. So, and in the end, we have pictures like these. Here you can see different markers that are known to be zonated. We see in green, which is a CYP enzyme, which is known to be around the central vein. The red one is the ecotherine. It's around the portal vein. And here we can see in blue, the vessels and the bile ducts are in yellow. So it was really possible to get the information of several proteins on one slide, which is, yeah, kind of cool. If you take a closer look, it looks like that. And by that, we were able to see more than we were able to see before with the normal IHC stainings. So here you see another staining. So we had the zonation markers here additionally in yellow, which is the glutamine synthetase, which is very narrowly around the central vein. And this is a bit closer. So we took a look at, does this zonation change? And we did not saw that it's changing, but it's only 3 markers. And we have to screen many more to get a better overview on that. But the most interesting thing is we looked at the proliferation in combination with these zonation markers. So here you see, again, the zonation markers, the ecotherine around the portal vein. We have here the central vein with the glutamine synthetase and the CYP enzymes. And additionally, we have in cyan, we have the phosphoristone 3, which is a marker for mitotic cells, and also PCNA, which is a marker used for S-phase entry, but is present during a lot or during the entire time of the cell cycle. And by that, we were able to see that if we take a look at the zonation, we see that in red is the portal cells, so the cells around the portal vein. We have the gray ones, which are the midlobular cells, and they enter the cell cycle a bit earlier than the pericentral cells. So we saw here a delay of the pericentral cells to get into the cell cycle, and also for the mitosis. So this was for the PCNA, but also for the phosphoristone 3. Then if we looked at extended hepatectomy, we saw, as we expected, less cells entering mitosis, but also a little bit of delay. So these cells started later in the cell cycle. At the moment, we're finishing also time points in between, so we get a better insight what happens in between. But what is the connection to the metabolic functions? So we know some of you have seen these slides already. This is also from a former lab member. One very important metabolic function is the beta oxidation. If you take a look at the standard hepatectomy, we see that we have this transient accumulation of fat in the liver. So as soon as the regeneration starts, the fat is accumulated in the liver. We see that, again, the arrow is lost here. We see an accumulation of fat, which is used for the beta oxidation, and we don't see it in the later time points. In an extended hepatectomy, where the regeneration does not work and we have a delay and we don't have an entry into the cell cycle, we see that this fat stays in the liver. It's still there after several days. And in, as I said, 20 to 30% of the cases of the mice, they will die. Beta oxidation is said to be the fuel for the liver regeneration, as we can see here, for several proteins which are important for beta oxidation, where we can see that the blue ones are the standard hepatectomy, they are increased. So we have an increase in beta oxidation, but we need to see it. And the problem is, what is usually done is you do a Western blot and you just take all the cells together, you mesh them and you measure them. And probably you already see what could be the problem. We have here a difference. So this is after sham operation, so only a cholecystectomy. And there we have the situation 2 days after the hepatectomy. And we see we have a difference. We have very upregulated single cells. But in a bulk analysis, this would get lost within all the other tissue that is not upregulated. And with fluorescence, we can see this. There are other methods to make like this single cell analysis, but fluorescence is a very good tool to get an insight into this. Then one thing I mentioned before is the hypertrophy. So cells that get big but don't divide. Here, maybe I explain a bit, is a resting liver, so a liver that has not done anything. And we see we have the red part, which is the periportal marker, the E-cathepsin, and we have here the central vein, it's here. And if you take a look, you see that after hepatectomy, we have an increase in size of the cells around the central vein. And we were able to see that in many different tissues. It's mostly between 32 and 48 hours after hepatectomy. And if we measure the size, and we have measured many, many hepatocytes by hand because there are not really good algorithms to do that by computer. We see that we have a massive increase in size of the cells which are located around the central vein. So we have a difference. We have a zonation of the proliferation and of the cell size after hepatectomy. But why and how does a hepatocyte know what to do? Why does it know, oh, I need to proliferate, or, oh, no, I need to get hypertrophic and probably do all the metabolic functions? And here we concentrated on P27. P27 is a cycle-independent kinase inhibitor, which can block the progression through the cell cycle at several points through the cell cycle. This is contrary to P21, which is known to block at one certain point. P27 is able to stop the cell cycle more or less at every point. It is regulated mostly by phosphorylation and ubiquitination, and not a lot is known about P27 in the context of liver regeneration. There is some studies about liver tumors, about tumors in general, because P27 can block also the cell cycle, and the downregulation of P27 is known to be associated with a proliferation or increased proliferation, which is seen in tumors. But most of this research is 20 years old and was never, people never got back to this. But we know that we need to lose P27 to get through the cell cycle in the end. And this is maybe a little bit a busy figure, but what you can see here is we have the pericentral cells, and we have the percentage of nuclei that are positive for P27. And we see that we don't have a significant difference, but if we take a look at the midlobular hepatocytes and also the periportal ones, we see that at 2 days after hepatectomy, we lose these P27 positive cells. So they lose it in order to get into the cell cycle and to go through the cell cycle. But here, we don't. And this could be a reason why these cells stay big, they don't divide, they don't go through the cell cycle. So we can also measure this. This is preliminary data because we have other methods to better measure this, but this is, at the moment an ongoing project. I will come back to that. What we can see here is that, in fact, we have the cells which are P27 positive are much bigger than the cells which are not P27 positive. We also see that the cells which are big or considered to be big have more P27, which is also a sign, okay, P27 is connected to hypertrophy. And if we connect this, for example, to HADBA, which is an important protein for beta oxidation, we see that if we take a look at every single cell, and here we see the p27 positive nuclei, which are yellow, and the green cells are the ones that are currently doing beta oxidation, we see that cells that are p27 positive, which we know are the big ones, are also the ones that are hypertrophic, are bigger. And this is more or less the start of the connection between the hypertrophy and the metabolic functions. So we know that this p27 is spatially and also temporally organized during the liver regeneration. We know that it is associated with hypertrophy. It is known to block or to be able to block the cell cycle, and we know that we need to lose it in order to get through the cell cycle. But what are these hypertrophic cells? Are they just big because they have these metabolic functions, or is there something more? And if you take a closer look at this picture, we see a normal liver, but we also observe that we have many cells with two nuclei. And these are the binucleated cells. So they have two nuclei. A lot of hepatocytes in our body, I think it's about more than half in the adult liver, is polyploid. So it has more than one nuclei or more than one set of genetic information. Also this hepatocyte, it's huge and it is assumed to be a polyploid hepatocyte. So why do we have this? This is not really known at the moment. But what do we know? We know that also the ploidy in the liver is zonated. So we know that most of the midlobular hepatocytes have a higher ploidy, have more genetic content compared to the ones next to the portal tract and to the central vein. This actually complicates this regeneration stuff a bit. What is known, or what other groups have found, that we have a change in ploidy during liver regeneration. We have a change from cellular to nuclear ploidy, so we don't have these binucleate cells, but one big cell with one big nucleus. We don't know if this is the only reason for hypertrophy, but one connection to our hypothesis that the big cells are also the ones that are metabolically active is that if you have more DNA content, you can also have more protein synthesis. So this is kind of an outlook of what I'm currently doing and how I try to solve this problem of single cells and how to analyze them. So the thing is, we can look at the tissue, but we can also try to isolate the cells. So take all the hepatocytes and get them into a single cell solution to do flow cytometry, for example, or to sort them by zone. And this is the setup. I think some people in our lab has tried to do this before, has done this before, but yeah, it's a bit tricky as the hepatocytes are really keen on dying. So as soon as you look wrong at them, they tend to die. How do you do the liver perfusion? You can cannulate the inferior vena cava or also the portal vein. I found that perfusing through the vena cava is much easier than the portal vein, and you can kind of retrogradely perfuse the liver. And this is how it looks like. So we have here in the beginning the liver, then it's cannulated, the perfusion solution goes through the liver and the liver swells, and all these hepatocytes are dissociated from each other. Then you have to carefully release the hepatocytes from its capsule, and then you are able to see the single hepatocytes in solution. And now comes the interesting part, you can try to analyze them. So this is some preliminary data. I was able to isolate them, to sort the single cells out, to take only the live cells, and to get rid of the non-hepatocytes. Because in the solution you also have endothelial cells, you have immune cells, and for the analysis you want to get rid of that. So we have here a more or less pure hepatocyte population, and now I looked at, for example, the Ploidy state after hepatectomy. So on top is the sham operation, we have here the state after hepatectomy, and we see that this is a dye, it's called Höchst, and the more DNA content is in there, the higher or the more to the right is the peak. So we have here the diploid cells, which are the normal cells. We have the tetraploid cells here, this is the peak, and we have also an octoploid peak. And what we saw is that there is a little difference. So we have an increase in ploidy after hepatectomy, and this needs to be further explored at different time points, maybe earlier, also later, but this is the preliminary data of that. But it seems that the standard hepatectomy induces the ploidy. Now, and this is the interesting part, we can analyze this by zone. So we have two markers which are contrary to each other depending on their location on this portocentral axis. So we have CD73, which is expressed around the central vein, and we have the ecatherine, which is expressed on the other side from the portal vein. And depending on their relation, we can say, this hepatocyte is from the portal vein, or this is from the central vein, or somewhere in the middle. And if we take a look at the ploidy profiles, we see that we have more or less similar ploidy profiles for these zones in the resting liver, but at the moment, we still don't know what happens after hepatectomy at these zones. And the idea is to get these cells also sorted. So we not only see the cells, but we can sort them in different tubes and analyze them afterwards. So this is kind of what we are going to do next. We are trying to analyze these ploidy profiles also in combination with proliferation markers, with metabolic targets. And in the end, we would like to sort them and do transcriptomic and proteomic analysis and also metabolomics on these sorted cells. Yeah, this is just a short overview. So we have here, usually we should have cells that go through S phase that get two cells in the end. But we know we have polyploid cells, or we get polyploid cells. We assume that P27 is important for that, so we block the cell cycle, that these cells get polyploid, and these are the big ones, which have more RNA, more protein, more metabolic functions, we assume, and are the ones which are really big. And by that, we'd like to connect the hypertrophy with the metabolic function, and also this nuclear ploidy, a story which kind of developed during the course of my experience. So thank you very much for your attention. Sorry for the overtime. Do you have questions about this?
So thank you, Eva, for this nice presentation and very clear overview of your project. And please, if you have any question, and I think Professor Clevin also like to join here.
Eva, thank you very much. I think this is a fantastic example what a surgeon can be, because you're a surgeon. How long have you been in the lab?
Now for a little bit more than 2 years.
Imagine, 2 years. That's not too much. How much clinical you've done before?
Before that, I think 2.5 years.
Okay. So this is what, I think we want to convince, and we are convinced, many of you here have done the same, that surgeon can do exceptional research for many reasons. The success is not just the surgeon doing it. As you have seen Anurag here, they are PhD. You can see how sophisticated is and becoming more and more this kind of research. The surgeons stay in the center because we have clinical questions. We see the patient, we operate this patient, and we see some of this patient dying. So we have the question. We know it's relevant. You go to the lab. Now today is different than for 20 years. You need collaboration, like the hepatocyte. They collaborate one with the other one to survive. We need to do the same in the academic world, meaning that we recruit PhD, pure biologists who are here. We have 3 in our department who will mentor or help the quality of the research. And we take resident, at least we select them, not in the past because they have no job, but we select because they are exceptional and potential. So going in the lab is we believe, demonstration that the good people we may have. And they don't go in the lab on Saturday evening. Many places in the world in surgery, or you do research on Sunday in the evening, etc. You can do some clinical work. Serious or relevant cannot be done that way. It's impossible. You take 5 years, 3 years to learn surgery. You take the same or more to learn some research with mentorship. So it's totally protected time. They may or may not do a PhD. They choose. I mean, we are not. They can go 3 years in the lab and just focus on a topic, or they can at the same time, and more like that, and they do a PhD with the basic science, so they can also learn statistics and other things and not just focus on the work. And then they come back, so they do these very relevant questions. This is important. We can change the way we treat these patients. And then hopefully you have also publication. It's not the only goal, but we have, and some of you have absolutely top publication, including in Science and other journals, that we cannot do alone as surgeon, but with the relevance and the help of basic scientists, we can sometimes achieve very, very important research. Then they go back because, Eva, what do you want to be in the future?
A surgeon.
Well, so she wants to be a surgeon. So she will go back and not be penalized in many places. 3 years in the lab, they come back and say, well, what are you? You're not a surgeon. Wrong. We have a fast track. We try to train them. And many of the staff here have been in the lab for 3 years, sometimes in complete different area, not automatically liver, but they can do colorectal. Doesn't matter so much because what you learn here is to think, to ask questions, to have hypotheses and to answer it. And that is also the same when you treat patients. So for your own life, and we have several surgeons that now in not an academic center, in other hospitals did this kind, and I think they benefit all the life of having been in the lab, and a few of them, I hope, I mean, I'm no worry that the future of surgery is great if we have a number of these people. And that's why we try here to fight, and not so many centers in the world, there's few in the US, who do that. That was initially this kind of program from Toronto and Johns Hopkins. They were that. They have this scientific program for residents, going 3 years in the lab, coming back, etc. And this is something that we believe in. Eva, I mean, I congratulate you. You are an absolutely perfect example. And I think you have a family too, right?
Yes.
So everything is possible. She has several kids. I don't know how you do it, but you do it well. And I think the demonstration that you can succeed with this if you have the right setting. So maybe I ask the first question now, because I just wanted to do that. 2 questions. This cell who basically they become big to work harder, right? To help the other one who go in mitosis and then will regenerate. That's what the regeneration would be at the end. So that's really good example of biology and collaboration. This hypertrophic cell, when they've done the job, they just, we don't know probably, they just go back to the normal size, or they go to mitosis too?
They actually, the thing is that most research about liver regeneration is done until a certain time point. And after that, we never looked back at the tissue. We know that the cells get hyperploid or polyploid, and we know that they go back. So if we consider a polyploid state somewhere kind of stuck in the cell cycle, we know that the cell can go back. So a hepatocyte, which is polyploid, can go back to the diploid state, but this will happen much later. So it's the hepatocyte has this potential to stop the cell cycle at every point and just go back to it and finish it later. Yeah, they're very plastic.
We have this ex vivo perfusion that we do in the VIS project when we can have liver for 10 days. And here we may maybe learn, because we cannot in patients, if you go to patient, do biopsy all the time. So, because that need to be translated to human liver. And the question as a surgeon, so now you did a lot of research, we are learning a little bit the failure, where are the mechanisms, what can we do for patients? How would you change something that maybe you prevent or can treat the small for size, which is a real clinical problem?
Yeah, this is a very good question. And if I would be able to answer this correctly and be sure about this, then...
But you can speculate.
My speculation is that we actually don't need more cells that divide firsthand, but more cells that do the job. So we need more cells that are big, hypertrophic or polyploid or whatever is behind this to get the job done, to relieve the system or the metabolism from the toxic stuff. So the problem is if a cell is dividing, it has no, at the moment where it's dividing, it's stuck, it cannot do metabolic functions. So probably it's better to kind of stop the proliferation a bit, to get the hepatocytes working and then to slowly restart it.
So that's a hypothesis to
test. It's a hypothesis.
So you have to test. Any questions?
I was very impressed by your talk. It was great. I have a question. I like your concept of you need more cells doing their job and not proliferating. Do you think that you could use an anti-proliferative drug at the beginning when you, after resection, like, I don't know, mycophenolate acid, where you stop the proliferation and have more cells doing their job?
Yeah, this is actually the idea, and I'm currently working on several inhibitors that are doing this. But probably it's wrong, but we have to test it. So,
All right. So I think,
thank you.
Thank you so much.
Thank you.